Thanks so much. Yes, so I was actually born in Oregon, and this is my first time back to my home state. It's beautiful. So thank you so much for that introduction. So I started my research laboratory at the University of Pennsylvania a little over 10 years ago, with the rationale being that if we could better understand the immune cells that cause disease in a rare but well-defined disorder such as Pemphigus vulgaris, that we could ultimately develop better immune-targeted therapies for that disease. So it's a real pleasure to give tonight's state-of-the-art lecture on, to highlight some of the research that's been done in that field to develop targeted immunotherapies. All right, I have no specific financial conflicts of interest to discuss regarding tonight's talk. I am an inventor on a licensed patent on work that will be outside the scope of the talk, and all treatments that we discuss will be off-label since nothing is currently FDA approved for Pemphigus. So I thought it would be interesting to kick things off by looking back to see what the state of the art was in 1965 for Pemphigus, which is a potentially fatal autoimmune blistering disease of the skin and mucous membranes. So Walter Lever writes in the introduction to his book, Pemphigus and Pemphigoid, the purpose of writing this book has been to bring up to date my monograph on Pemphigus that I published in 1953. Actually, no fundamental discoveries have occurred in the field of Pemphigus during the decade following the publication of my monograph. So I had to laugh because they always say that scientists are terrible at selling their work, and this is a really extreme example of that. It doesn't exactly make you want to turn the page, but if you do, what you can find is that back in the mid-1960s, we had no idea what caused Pemphigus. There were theories on salt retention, viral origin, enzyme genesis. One of the examples he gave is that trypsin also causes keratinocytes to fall apart, so perhaps something similar was contributing in Pemphigus. So it was actually just before the publication of that book that Beutner and Jordan discovered that Pemphigus is in fact an autoimmune disease, which they published in a series of studies, including one ultimately published with Walter Lever, describing the direct immunofluorescence and indirect immunofluorescence tests for Pemphigus that are currently in clinical use today for diagnosis. And in there, they documented the presence of anti-epithelial IgG on the surface of keratinocytes, both within the skin and also in the blood of Pemphigus patients. And over the next quarter of the century, Pemphigus, the molecular targets of those autoantibodies would ultimately be discovered. Pemphigus foliaceus antigen as desmoglein 1, and Pemphigus vulgaris antigen as desmoglein 3 by Lina Kulu and Masa Amagai working with John Stanley. And in the last quarter of a century, Pemphigus vulgaris has gone on to be established as arguably one of the best defined autoantibody-mediated diseases in humans. So disease is actually relatively straightforward to diagnose. There's a very classic pathognomonic suprabasal blister that we observe on histology. Of course, the cell surface immunostaining that we see on immunofluorescence. And anti-desmoglein antibodies are 98 to 100% sensitive and specific for disease diagnosis in Pemphigus vulgaris based on the commercial ELISA assays. Additionally, anti-desmoglein 3 antibodies are both necessary and sufficient for blister formation, even as monovalent antibody fragments, which we think occurs due to their direct effects on steric hindrance of desmosomal cell adhesion, interference with desmosomal trafficking, as well as initiation of cell signaling pathways. And that research, which I don't have time to cover today, has been elucidated by a number of great research teams here in this audience as well as around the world. So we've come a long way in the last 50 years in regard to understanding the basic mechanisms of disease. But if we go back to the 1960s and we look to see what they said about treatment, we're pretty much in the same place. The importance of administering corticosteroids in massive doses during the initial phase of treatment has been generally recognized and has led to a great improvement in the long-range results. And that pretty much summarizes the current state of treatment today in 2017 for Pemphigus vulgaris. It's all about balancing the risk of disease with the risk of therapy. It used to be that Pemphigus vulgaris was fatal without therapy. Now that we have immunosuppressive therapies such as prednisone, mycophenolate, azathioprine, and rituximab, which globally suppress or eliminates parts of the immune system, patients are no longer dying from their disease, but they're suffering from complications of its therapy. So for example, chronic immune suppression with rituximab is associated with a 1.3 to 1.9% rate of fatal infections in meta-analyses and national registries, among other side effects. So really, the ideal for pemphigus therapy, as well as all autoimmune therapy in general, is to target only the pathogenic autoimmune cells and spare the rest of the good immune system that helps to protect you from infection. So because antibodies are produced by B cells, B cell forensics became the focus of my laboratory when I first started it. There were so many things that we didn't know about pemphigus patients. We didn't know how many different anti-desmoglein B cells are there in pemphigus patients. Are there shared features of B cells among pemphigus patients? Do they have shared antibody VH genes or amino acid sequences that we could ultimately target? So the patient's blood became the clues that we could bring back to the laboratory to try to figure out what caused their disease. The approach that we took was B-cell repertoire cloning to isolate and characterize the anti-desmoglein 3 B cells from Pemphigus vulgaris patients. In one of the approaches that we took Pemphigus patient B cells, we PCR'd up all known antibody heavy and light chain genes to create an antibody phage display library. We then selected those clones against desmoglein 3 to identify individual anti-desmoglein 3 monoclonal antibodies that we could ultimately produce as soluble antibodies for testing. In another approach, we took pemphigus B cells and we fused them with mouse myeloma cells to make antibody-secreting heterohybridomas. We screened their supernatants against desmoglein 3 and used limiting dilution cloning to, again, isolate individual anti-desmoglein 3 heterohybridomas from pemphigus patients. And ultimately, by having access to both the genetics of the B cells as well as the antibodies that they produce that we could perform functional pathogenicity testing on, in that way, we could actually perform an in-depth genetic and functional characterization of the human anti-desmoglein 3 B cell repertoire. So the B cell repertoire cloning that we have done now from multiple PV patients, the first 5 of whom are shown here, has identified multiple desmoglein 3 reactive B cell lineages. Because we have the sequences of these antibodies, we can ultimately arrange them into pedigrees. Each circle is an individual B cell clone. Which represents a distinct B cell lineage from distinct B cells that came from the bone marrow. And all of the derivative branches are daughter B cells that have somatically mutated or affinity matured away from that parental B cell clone. So if we delve deeper into the genetics of these B cell lineages that we've identified, the thing that immediately jumps out at you is that anti-desmoglein 3 B cells from all 5 of the Pemphigus vulgaris patients that we looked at use VH146, a particular antibody VH gene. Also others were identified, but only VH146 was shared across all 5 patients. The reason this was intriguing is that Masa Amagai had generated an active immune mouse model of PV, where mice developed mucosal erosions and patchy hair loss, as well as hair follicle acantholysis, similar to what was observed in human PV patients. And the major pathogenic clone that they identified from that mouse model, AK23, uses murine VH153. If you take the amino acid sequence of murine VH153 and blast it against the human antibody database, its closest match is human VH146. So in both mouse and man, anti-desmoglein 3 B cells appear to favor VH146 B cell usage. So what makes these VH146 B cells so prone to bind desmoglein 3? An interesting feature when we looked at these VH146 B cell sequences is that the anti-desmoglein 3 antibodies had relatively few amino acid mutations. If you look at a B cell coming out of the bone marrow, it's called naive, it's never seen antigen, and it has no mutations reflecting its inexperience. But after it is exposed to antigen, it travels to secondary lymphoid organs where it activates DNA mutating enzymes such as AID, and repetitive mutation as well as selection for higher affinity binders then leads to a characteristic pattern of mutations in memory B cells, namely a median of roughly 20 mutations and an increased ratio of replacement to silent mutations in the portions of the antibody that contact antigen, which is also known as the CDRs. As opposed to the framework regions in which the replacement mutations are relatively selected against because they can disrupt antibody structure. So the fact that we find relatively few mutations in the VH146 B cells then begs the question, do those VH146 B cells even need their mutations to bind to desmoglein 3? Because we have the mutated sequences of the antibodies and we know their germline sequences, we can take each of the somatic mutations that we identify and revert it back to the germline amino acid residue and ask, do those germline reverted antibodies bind to desmoglein 3? Essentially asking, was that B cell desmoglein 3 reactive in its naive and unmutated state? And what we found was that 3 of the 5 VH146 lineages we identified did in fact recognize DSG3 in their unmutated state. So the parental somatically mutated antibody is shown in the solid line, and all of the germline reverted versions are shown in the dashed line. So these 2 did not recognize, these 3 did, which we confirmed by staining of human skin with a characteristic suprabasal pattern. And even this particular clone, which showed some weak binding by ELISA and undetectable binding by immunofluorescence, had a 10 to the negative 7th molar affinity by surface plasmon resonance, which is well within the range that's required for B cell activation. In contrast, if we looked at B cells using all other VH genes, none of them bound to desmoglein 3 in the unmutated state. So all of these were negative. This particular clone, we only reverted back the light chain mutations. In a series of studies that aren't shown here, we showed that really it's mutations within the heavy chain of these antibodies, not the light chain, that determine that specificity. So really these data get to when and where does B cell autoimmunity to desmoglein 3 develop? So for VH146, we think that rare B cell clones emerging from the bone marrow may weakly bind desmoglein 3 due to residues in and around the CDR2 region of the antibody that favor desmoglein 3 binding. That was work that was done that I didn't have time to discuss today. And somatic mutations later in life may lead to higher affinity antibodies that are disease-causing. But because VH146 B cells are a little bit prone to bind desmoglein 3, that's why they're more commonly found. In contrast, B cells using other VH genes are generally not born desmoglein 3 autoreactive, but somatic mutations can create newly desmoglein reactive clones in the periphery, some of which are pathogenic. So these data gave us hints into the immunopathogenesis of disease, the fact that we find common humoral immune responses in Pemphigus patients. But how can we use this to develop targeted therapies? So in one of our earliest papers, we explored the concept of VH gene-targeted therapy for PV. The idea here is if you take the variable region of a PV anti-desmoglein 3 monoclonal antibody, inoculate it into rabbits, isolate the antisera, and clear it against normal human IgG to clear it against antibodies that react against framework and other portions of the antibody, you can purify anti-idiotype or VH gene-targeted reagents. And what we found was that if we generated a mixture of anti-IDs against the VH genes that we identified from one particular patient, it would actually work to block the pathogenic activity of the PV sera from that one patient. But if we tested that mixture of anti-IDs against other PV patients, it wasn't sufficient to block all of their pathogenic activity. And that perhaps makes sense because, of course, each particular PV patient is different in regard to the different VH genes that comprise their entire anti-desmoglein and autoimmune repertoire. So people were always saying, you know what causes Pemphigus, it's desmoglein, why don't you just use desmoglein to target? And in fact, our collaborators have done that. So Masa Amagai's group, again, has conjugated desmoglein 3 to Pseudomonas exotoxin, the idea being that desmoglein 3 chimeric protein can then bind to anti-desmoglein 3 B cell receptors on the surface of memory B cells. They then get internalized into the cell, the toxin goes to the nucleus, and it ultimately can kill that B cell. Ultimately, this had limited efficacy in vivo. It could have been for a variety of reasons, including the efficiency of toxin-mediated killing, the half-life of the desmoglein 3 chimeric protein in the serum, and also the fact that desmoglein 3 chimera can be one-to-one neutralized by soluble serum antibodies. The research group from Euroimmune has proposed desmoglein 3 specific immunoadsorption as a potential treatment for Pemphigus. So unlike general immunoadsorption, which results in just removal of all of the IgG in a patient's plasma, the use of a desmoglein 3 column would specifically absorb out only the anti-desmoglein 3 antibodies from a Pemphigus patient's plasma. So of course that would be very targeted. However, because it doesn't stop the antibody production, ultimately repetitive treatments would be necessary to maintain disease remission. So recently, rituximab has emerged into the spotlight as a very effective means for Pemphigus therapy. Rituximab is an anti-CD20 monoclonal antibody originally approved for non-Hodgkin's lymphoma and other B-cell cancers. In a recent study published, when used as first-line therapy with steroids, rituximab leads to an 87% rate of complete remission off therapy after a median of 9 months. And even more interestingly, if we look at the anti-desmoglein antibody titers after rituximab, the titers drop to the normal range. If you focus just on the one in solid line here, it actually drops below the detectable range, meaning that rituximab is sufficient to deplete the key pathogenic population, that anti-desmoglein antibodies are predominantly produced by short-lived plasma cells that are highly dependent on that CD20 positive B-cell pool. So this exciting data led to rituximab receiving breakthrough therapy designation for the treatment of Pemphigus vulgaris just within the last month. However, we do know that the majority of patients that are treated with rituximab ultimately relapse and require repetitive treatments to restore or maintain disease remission, which then causes chronic immune suppression and all of its associated morbidity and mortality. So at this point, enter chimeric antigen receptors, stage left. In the mid-2000s, Penn was really abuzz with Carl June's groundbreaking work using chimeric antigen receptors to treat relapsed and refractory B-cell leukemias. A chimeric antigen receptor consists of an antibody-based extracellular domain which can direct T-cell cytotoxicity with high specificity against any target. In the case of B-cell leukemias, it was CD19. It has a T-cell receptor cytoplasmic signaling domain, and upon binding antigen, CAAR-T cells will expand to kill their target cell through activation of that T-cell. And all CARs also have a co-stimulatory domain, or signal 2. In the case of the Penn construct is CD137, which appears to confer greater CAR-T persistence, perhaps more so than other CAR designs that are in clinical development. So this work was done, of course, not only at Penn, but by many institutions around the country, including Memorial Sloan Kettering, NIH, Fred Hutch, Baylor, and many others. And how does CAR-T work? We collect the peripheral blood mononuclear cells from patients undergoing apheresis, activate the T cells, transduce them with lentiviruses that gene engineer them to express these CARs, proliferate them, and then return them back into the patient. And what happens when you do that is that in the pivotal phase 1/2a trial of 30 patients with relapsed and refractory ALL seen at Penn, that 90% of them went into complete remission and 73% of them relapse-free. The major toxicities observed were permanent B-cell depletion. These CAR-T cells engraft and potentially persist for the lifetime of the host, causing lasting protection from cancer recurrence, but also lasting depletion of those B-cells. And most of the patients experience a cytokine release syndrome, which is a massive immune activation that occurs from CAR-T proliferation and leukemic cell death. So despite these toxicities, the fact that 73% of patients who otherwise faced certain death were now in relapse-free remission from their disease was really a phenomenal accomplishment that led to CTL19 receiving FDA breakthrough therapy designation for the treatment of ALL. And it also brought Joe Biden to Penn Med to launch his moonshot mission to cure cancer, because we always say that we never use the word cure when it comes to cancer, but they felt that a cure was finally potentially within our reach. So enter Christoph Ellebrecht, stage right. Christoph Ellebrecht is a really passionate and brilliant physician scientist from Germany who I was really fortunate to have join my laboratory several years back. And if we think about treatments for leukemia, we think about that a lot because, of course, rituximab, which is one of our most effective therapies, is used to treat B-cell leukemias. And really, a lot of the things that CAR-T cells were being used to treat relapsed and refractory cancer sounded a lot like our Pemphigus patients, relapsed and refractory Pemphigus. So the CD19 CAR consists of an anti-CD19 antibody that targets the CD19 antigen on B cells to cause lasting B cell depletion. But in thinking about how to re-engineer CAR-Ts for autoimmunity, those same B cells, in the case of the desmoglein reactive B cells, express an anti-desmoglein 3 B cell receptor on their surface, essentially marking the pathogenic autoimmune cells exactly the ones that we want to target. So in this case, Christoph used the autoantigen itself, desmoglein 3, as the extracellular domain of the chimeric immunoreceptor to specifically target the anti-desmoglein 3 B cell receptor on CAR-T. CAR-Ts. So he befriended an instructor in Mike Malone's lab who had helped to develop anti-CD19 CAR-Ts for leukemia. And I guess the rest, as they say, is history. The chimeric autoantibody receptor, or AA CAR, as we like to call it, came into being. So the design of the desmoglein 3 CAR turned out to be important because it has to fall within a certain size limit. Desmoglein 3 naturally has 5 extracellular cadherin domains. We know from epitope mapping studies that EC1 through 3 are most often targeted by PV autoantibodies. So we developed a series of constructs for the extracellular domain of the desmoglein 3 CAR. And a priori, we didn't know if T cells would be able to express them. Desmoglein 3 is a pre-pro protein that has to be cleaved in a particular way to remain conformational. And what we found was that EC1 through 3 and EC1 through 4 CARs were nicely, robustly, and reproducibly expressed on the surface of CAR T cells, whereas EC1 through 5 was really quite variable. Here's its expression across multiple experiments, and it really didn't ultimately show effective cytotoxicity against anti-desmoglein B cell targets. So we needed to determine whether these desmoglein CAR T cells could specifically kill anti-desmoglein 3 B cells in vitro. And so for this, we selected a variety of anti-desmoglein 3 B cell targets informed by the ones that we had cloned from Pemphigus patients, as well as those that have been isolated from the active immune mouse model of Pemphigus. AK23, 19, and 18 are mouse hybridomas that are B cell receptor surface positive and also cross-react to human desmoglein 3. They recognize different EC domains, and they have a broad range of affinities against human desmoglein 3. Similarly, F779 and PDB28 are human anti-desmoglein monoclonal antibodies isolated from PV patients that recognize EC1 and 2. So to do these assays, we load up the target cells with chromium, and we calculate a percent specific lysis relative to an SDS lyse control. And so just showing one of these for the purposes of simplicity, if we look at the desmoglein EC1 through 4 CAR, we find that it very specifically kills AK23, 19, and 18 anti-desmoglein 3 B cells, but it doesn't kill a control hybridoma expressing a B cell receptor of an irrelevant specificity. And conversely, control CAR T cells don't kill the anti-desmoglein B cells, indicating the specificity and also the potency of the approach. To preclinically validate the human desmoglein CAR T cells in vivo, we decided to go with a PV hybridoma model. For this, we can inject a polyclonal mixture of hybridomas that we know secrete anti-desmoglein antibodies. It causes mucosal blistering and results in circulating and tissue-bound anti-desmoglein antibodies that parallel the findings in human patients. Again, it also allows the sensitive tracking of B cell elimination because we can bioluminescence label the hybridomas that we inject. And it also allows us to carefully evaluate the success of the CAR-Ts in eliminating B cells of a broad range of epitope specificities and affinities, both as a mixture and individually. And also, it allows testing of the human desmoglein 3 CAR-T cells that would ultimately be necessary for an IND application. So we injected a polyclonal mixture of these anti-desmoglein B cells at day 0, allowed them to grow logarithmically, and on day 5, injected either desmoglein 3 CAR T cells or control CAR T cells and serially followed them with bioluminescence imaging and serum antibody analysis. And what we found was that the control CAR-T didn't have any effect on the hybridoma outgrowth. They continued to proliferate logarithmically. In contrast, the desmoglein 3 CAR successfully controlled and in fact eliminated by bioluminescence imaging the desmoglein 3 surface antibody positive CAR-T cells. If we look at the serum of these mice, what we find is that desmoglein CAR-T treatment reverses the rising serum autoantibodies that were seen at baseline to an undetectable level by 9 days later, whereas in control CAR-T mice, the serum antibody titers continued to worsen. Ultimately, this resulted in a reversal of the serum autoantibody binding to mucosal epithelia, as well as the blistering that was observed in these mice. So ultimately, the mice were both histologically as well as serologically in complete remission. One thing that we weren't sure what to expect a priori was what the presence of soluble anti-desmoglein 3 antibodies would do to the system. So on the one hand, the soluble antibodies could bind to the desmoglein 3 CAR T cell and neutralize it or cause its phagocytosis. But on the other hand, we know that when desmoglein CAR T see their target, they proliferate and expand. in response to target binding. So what we did was we initially set up a series of monoclonal assays where we asked the desmoglein 3 CAAR-T cells to kill their target cell in the presence of increasing concentrations of the exact monoclonal antibody that it could see, where we knew that that antibody was 100% neutralizing. And what we found was that we saw mixed effects. Some of them were inhibited, some of them really didn't seem to do anything, and some of them were in fact potentiated in their killing. And if we looked at polyclonal sera against CAR-T cells, what we found is that polyclonal sera actually stimulated a low level of interferon gamma production, shown here compared to, for example, a positive control, and also stimulated CAR-T proliferation. And although we didn't have enough data points to generate a firm conclusion, what we found was that antibodies that tended to go on and had a very high affinity with a very where they basically went on and stayed on, those were the antibodies that tended to inhibit CAR-T activity. Where the antibodies that went on but had a fast off rate were the ones that tended to stimulate and proliferate the CAR-T cells. So overall, taken together, we feel that because all patients will present a combination, a polyclonal mixture of those antibodies, that ultimately the polyclonal mixture likely won't prevent and in fact could help promote CAR-T engraftment and efficacy. So off-target toxicity is important. A priori, the one that we wanted to investigate most closely was the potential for human skin toxicity, because desmogleins and desmocollins are the natural ligands for desmoglein 3 in the body. So we made human skin xenografted mice. Even though this was one of our major targets we wanted to explore, we felt that perhaps we wouldn't see toxicity. Number one, because desmoglein and desmocollin is a low affinity interaction. It's only on the order of a micromolar level affinity, and typically the affinities that are required for good CAR-T efficacy are in the order of nanomolar to picomolar. Additionally, we know that the size of that intermolecular synapse is important for cytotoxicity. So ideally, the T cell needs to be about 15 nanometers away from its target cell to effectively kill it. And we know that the desmosome is about 35 to 40 nanometers in terms of its intercellular distance. So we felt that that was probably going to prevent effective CAR-T cytotoxicity. So these were the human skin xenografts on mice, and what we generated as a positive control was a PX44 or an anti-desmoglein 3 CAR to generate skin toxicity. And what we find is that these anti-desmoglein 3 CAR T cells really hone right into that graft. Here on a CD3 stain, you see that they infiltrate the epidermis quite heavily, and classic signs of keratinocyte toxicity and satellite cell necrosis were apparent. In contrast, our negative control, CAART19, which has been in human patients and is known not to cause direct skin cytotoxicity, didn't show any histologic evidence of cytotoxicity and only showed a sparse level of epidermal infiltration, which is consistent with allogeneic effect because our donor skin samples and donor T cells weren't matched. And the desmoglein CAR T cells showed that same level of background alloreactivity with no direct evidence of keratinocyte toxicity, either in this experiment or in a number of in vitro studies that we conducted. So in summary, decades of research to better understand mechanisms of disease in Pemphigus has created knowledge that has really enabled the development of targeted therapies for this disease. B-cell depletion is an effective therapeutic strategy for Pemphigus, but requires repetitive therapy with risk from immunosuppression. CAR-T cells represent a novel approach to targeted therapy of Pemphigus and may not be susceptible to some of the problems that we have with CAR-T therapy of cancer. So the reason we think that only 73% had lasting remission and not 100% is that B-cell leukemias can either shed or mutate CD19. In the case of an anti-desmoglein B-cell, we don't think that that will really be possible. If a B-cell sheds its B-cell receptor, by definition, it cannot be activated. It essentially becomes functionally anergic, so it can never be activated to secrete antibody. And additionally, if a B-cell mutates its desmoglein 3 B-cell receptor so that it no longer binds desmoglein 3, then that's also perfect because it by definition becomes irrelevant to disease. Additionally, cytokine release syndrome, we don't think it occurs because it's related directly to B-cell burden. And we know that anti-desmoglein 3 B-cells typically only make up about 0.1% of the total B-cells within a patient. So we hope that CAR-Ts will offer the potential for lasting remissions of Pemphigus and could ultimately serve as a universal strategy for any antibody-mediated disease. So I really want to thank everybody that did this work, including our collaborator, Mike Malone, who helped develop CAAR-T, and of course, most importantly, Christoph Ellebrecht. And I encourage you to go see his talk, which is at the Saturday plenary, talking about temporally controlled CAR-T as an alternative strategy for pemphigus therapy. So I wanted to thank my funding agencies as well as the Dermatology Foundation, which has supported me throughout my career. Thanks so much.